Hey Megan, want to hear a joke?
Yeah, sure.
What type of jokes can you tell during quarantine?
What type?
Inside.
Oh my god, we're recording.
Hey guys, welcome to Plant Rant's news channel. This is where we rant about plants.
So in today's special broadcast, we'll be talking about oak apple galls. I'm sure you guys are wondering what these even are, so we're here to explain all about them.
So, this is an oak apple gall. And as you can see, it looks just like a normal plant. However, this is not the case.
Oak apple galls are actually not any ordinary plants. They're in fact large, irregular plant growths which are stimulated by the reaction between plant hormones and powerful growth-regulating chemicals produced by some insects and mites.
Yes, these plants in fact are created by insects. Isn't that crazy? Well, we will have our expert Harleen tell you about that in just a little bit. But first, let's continue and tell you all about this amazing plant growths can range in size from half to 2
inches in diameter. These galls are constructed of leaf tissue that has been hijacked by a gall wasp to surround a single wasp larva, located within a seed-like position at the center of the gall. So in simpler terms, the wasp makes the gall to house its babies in, using the nutrients of the gall itself to nourish the larvae. Isn't that fascinating?
Definitely fascinating. The gall can also be used to protect the inhabitants from natural enemies along with pesticides. These galls can occur in many different places. They can be on leaves, bark, flower, acorns, roots, etc. Leaf and twig galls are the most noticeable. In particular, the oak apple galls most often grow on black, scarlet, and red oaks. They actually get their name for their resemblance to apples.
Definitely wouldn't want to eat that apple. Well, now that you guys know all about the general oak apple gall, let's go to our expert Harleen, who will tell you guys about how these oak apple galls are formed, in particular by the oak apple wasp.
Hi, this is Harleen reporting from Davis. You may be familiar with where I am right now. That's right, I'm in front of Olson Hall. And this tree you see over here is an example of a huge gall tree. However, for the case of our plant, the tumor is much smaller. It's only about 2 to 3 inches in diameter, just like this apple. The best time to find these galls is during the months May and June, when gall formation is initiated by a female wasp as she lays her eggs on oak twigs. The interaction between plant and wasp is symbiotic. The wasp uses the gall as their home. While the gall provides protection, the tissue inside provides nutrition. The gall maker directs tumor growth to suit their own needs. Let's look at a visual for better representation.
The inside of a gall is composed of chambers that house immature larvae. Depending on the gall maker, the internal tissue could have a mass of white fibers or have a spongy structure. Once the larvae mature, each housing larvae eats their way out and the oak galla changes appearance and shape as well. Mature galla become brittle and turn light brown. With no larvae to house, the filaments inside disintegrate, leaving the gall hollow and empty. While this was just a short brief on gall formation, the actual mechanism is quite complex. Let's return back to my colleagues so they could unveil the mysterious mechanism of apple oak gall.
We heard all about these wasps forming these apple gall oaks. We now are going into the particular mechanism and how this wasp is able to make these galls, and how they are able to take plant tissue and transform it into a home for their larvae. So we know that the galls are formed from various plant tissues depending on the host and insect involved. Gall formation by wasps and other insects is a very complex, fascinating process that has yet to be fully understood. In fact, no researcher has ever produced a plant gall without the aid of a gall maker. Isn't that crazy?
Yes, so crazy. So now you're all wondering, if this is such a complex process, how are we going to explain it to you guys? Well, don't worry. Research still has a general idea on this process. So the galls are formed from plant tissue in response to chemicals produced by insects, which mimic various plant growth hormones. The reason this gall-making process is so complex is because gall-making insects produce chemicals that turn plant genes on and off at just the right time to direct gall growth.
Yes, these gall makers are able to produce chemical replicas of plant hormones referred to as hormone analogs. These molecules that may be nothing even structurally similar to the plant hormones. However, the plant response is still the same as it would be.
So what exactly is the physical mechanism on how this formation starts? Well, it is initiated by the female wasp when she injects gall-inducing chemicals into the plant along with her eggs. These eggs may also ooze gall-inducing chemicals, and once the eggs hatch, the interaction continues with the immature gall maker continuing to exude chemicals to direct plants' growth to suit their needs, which in this case, the needs are forming the gall.
Whoa, chemicals. Talking about chemicals. So, how do these chemicals that are exuded by gall makers make these galls? Well, first, these gall makers can only act upon meristematic cells. This is because these are undifferentiated cells that are yet to develop into a particular cell variant. Since these cells have not yet formed what their function is, this is perfect for gall makers to work on. Some of these gall makers actually exert their influence on the meristematic cells in leaf buds, precursors to leaf cells.
Under the influence of chemicals exuded by gall makers, the meristematic cells that were originally destined to become leaf cells begin doing what the gall maker wants. These errant leaf cells fall under the chemical spell of gall makers and become what these wasps want them to be, aka gall tissue. This is not a reversible process, and when these cells become influenced by the chemical produced by gall makers, they will become gall tissue.
Like Megan said, since these cells have become influenced by gall makers, they will become destined to make galls. So, in the opposite case, what this means is that the gall formation cannot form once the meristematic leaf bud cells are already committed to becoming leaf tissue. So, if an undifferentiated cell becomes destined to become a leaf tissue, it cannot become a gall maker. So, now that we know about the chemicals and how they work, what is the mechanism on how these are capable of turning off and on genes in the exact moment to form oak apple galls?
The initiation of insect galls is typically associated with oviposition by the adult gall wasp, or by the feeding of early larval stages. Depending on the type of insect and whether it has chewing, piercing, or sucking mouthparts, a variety of salivary fluids may be injected into the plant tissue to cause this initiation.
Saliva from bugs and aphids may include amino acids, auxins, and various plant digesting enzymes such as pectinases, cellulases, and proteases. The precise mechanism by which these chemicals induce cell division and morphogenesis to cause this gall is very complicated and varies with different species and even with different types of plant tissue. An increased concentration of certain plant hormones in the gall tissue may also be important for the morphogenesis of some galls.
Part of the reason the gall-making process is so complex is because gall makers produce chemicals that turn plant genes on and off at just the right time to direct gall growth. Many aspects of this interaction between the wasp and its host plant are poorly understood as the mechanisms of gall induction are still largely unknown by scientists.
Since even the scientists are unsure of the exact mechanism and genes involved in the gall formation process, we have constructed an example to display what we think a simplified version of this pathway may look like. Here we have the simplified mechanism of oak apple gall formation. This is induced by gall wasp chemical, which activates gene A, a positive regulator. When this A is activated, we can see it inhibiting gene B, and we see oak apple gall formation. However, in the opposite case, when gene A is not activated, gene B, a negative inhibitor, inhibits gene C, and therefore there is no oak apple gall.
Hey, welcome back. Now that we've talked about some mechanisms, let's look into a case study that was done right here in Davis. The study was to determine the association between wasp and oak apple galls. From what we've learned so far, we can tell that wasp and other gall inducers basically manipulate gall growth to produce structures to meet their own suitable needs. Although it is still quite unclear what specific responses occur, the study found that abundances of specific wasp species in gall could actually help us understand the pattern of gall development and size. Researchers examined different species of insects emerging from a variety of oak apple galls. Insects were grouped according to their morphology, and the most common species was determined by where the larvae had developed in the gall chamber. They analyzed the association of insect with gall traits first in the absence and then in the presence of insect species. They found that gall inducers A. Corcus californicus were more abundant in larger galls, while T. californicus were present in smaller galls. Well, there you have it. We could determine the size of the tumor by just looking at the gall-inducing species. However, while we have learned a great deal of wasp and galler relationships, the exact details of galla development are yet to be studied. I hope you had fun learning with me. Let's return back to Megan and Kanika.
Thank you, Harleen. Isn't it crazy how all of this can be going on around us right here in Davis and most of us have no idea?
Now, every time you walk by Olson and see that tree, you will be reminded by the phenomenon that is oak apple galls.
Thank you for tuning in to Plant Rants News, the news outlet with the hottest information on galls. We will see you next week for our broadcast on crown gall tumors.